A single expected path appears to define reproduction across most of biology. One narrow channel carries the entire narrative of how life passes from one generation to the next. But even early biological factors already push some flow aside, creating the first meaningful fork. Genetics and prenatal development introduce variation at the very origin. Developmental timing carves another stream entirely. The precise arrival of hormones, the sequence of growth patterns. Each of these factors adds a new tributary to the growing network. Later social experiences widen the network even further. Environment and upbringing write their own tributaries into the branching structure, redirecting the current in unexpected ways. Preference channels multiply across the landscape, while the original expected path narrows, gradually becoming just one possibility among many, rather than the single default route biology seemed to promise. The resulting pattern looks natural, organic, much like a real river delta formed over time. Yet the question at its center remains unresolved, openly unanswered. Are these branching patterns strictly human constructions? Or do other species share similar diversity? Do we observe this spectrum of variation across the animal kingdom? This river delta of sexual variation poses the very question that drives our exploration ahead, a question with no simple answer and no single channel. Why do many preferences lock in so early? The developmental timeline reveals a hidden pattern. Certain windows are far more sensitive than others to environmental input, creating lasting imprints. Prenatal hormones begin organizing reward sensitivity even before birth, shaping the brain's early response circuits for pleasure, motivation, and social bonding. In infancy, sensory mapping continues the wiring. Every sight, sound, and touch builds the perceptual foundation for later preferences, aversions, and emotional associations. During adolescence, reward circuits sharpen specific triggers. Synaptic pruning and dopamine pathway intensification narrow what the brain finds rewarding. Each critical window narrows the range of later adult patterns. The earlier stages remain structurally visible, but their plasticity fades and their influence becomes constrained. The map becomes more individualized with every stage. Each person's unique sequence of developmental windows creates a personalized brain architecture unlike anyone else's. The same sequence appears in other mammals too, from rodents to primates, suggesting a deeply conserved biological blueprint that evolution has preserved across species. How much of adult desire traces to these early maps? The question remains open, but the mechanism of critical windows shaping reward sensitivity is undeniably powerful. The early brain map is just a starting scaffold. Experience begins layering on top, shaping which connections survive and which fade away. An unexpected pairing can link a neutral cue to a reward. A single event creates a new association where none existed before. With repetition, that link strengthens into a neural shortcut. Each recurrence thickens the pathway, making it faster and more automatic. Mental rehearsal works similarly. Just imagining the loop activates the same circuits, further entrenching the pattern without real-world input. Positive feedback accelerates the process. Each successful reinforcement makes the selected route even more likely to fire again. Meanwhile, unused pathways lose their strength. Connections that are not reinforced weaken through disuse, while strong ones stay bright. The result is a highly individualized circuit. Experience has sculpted the general starting map into a specific adult pattern. But are these learning rules unique to humans? Animal studies suggest similar mechanisms shape behavior across species too. Is the entire spectrum of sexual behavior uniquely human, or does it share biological roots across mammals? Take primates. Non-reproductive mounting emerges after early pair bonding cues light up the same reward circuitry. Now look at humans. The same developmental cues trigger identical reward patterns and mounting behaviors. Dolphins show repeated same-sex behaviors driven by the same reward mapping. Social cues light up identical centers. And once again, the human side mirrors that exact reinforcement loop. The biological pattern is shared. Bonobos layer social bonding onto these circuits, while humans overlay cultural narratives. Same wiring, different topcoats. Strip away the species labels, and the mechanism is identical. Biology erases the boundary between animal and human. The only difference? Humans build elaborate cultural stories on top of that shared biology. That is the amplifier. Look at this delta now. Multiple mammalian species, from mice and rats to primates and humans, all feed into the same branching network. Every stream originates from ancient biological machinery. Beneath the surface colors, the shared river sections grow darker and thicker. This is the conserved mammalian wiring, the common base that every species inherits from a distant ancestor. But notice what happens on just one branch. The human channel receives an extra element, a cultural amplifier. No other species has this layer. Watch what the amplifier does. The human stream widens dramatically, expanding far beyond the narrow biological range. Culture amplifies variation. Meanwhile, every other species stays within the ancient mammalian range. Their streams hold steady, unchanged by the expansion happening right next to them. Now all streams curve toward a single destination. The delta fans merge into one broad ocean, a shared space of behavioral possibility that every mammal occupies. Above the merged sea, the cultural amplifier remains visibly distinct. It is the sole differentiator, the one addition that sets the human branch apart from all others. So here is the takeaway. Kinks arise from shared mammalian mechanisms, ancient circuits we all carry, yet they receive uniquely human expansion through culture alone.